So definitely as the last person here, I want to take this opportunity to thank James and Blanca, and I'm sure a lot of people behind the scenes for putting together a really wonderful conference with uniformly good talks, at least up until now. We'll see how this one goes. And I just want to say also that, you know, Spain is a remarkable country. So not only do you have a genomics center on the beach, but you also, you know, if you walk along parallel, you can get educated by the great systems biologist Ferran Adria, who has studied the genome of Spanish cuisine. And if you're lost in a little village by the sea, there's a sign that tells you where to go for omics. Right? So it's a pretty amazing place, and I've enjoyed my time here for sure. So what I want to tell you about today is my lab's work on non-genetic heterogeneity, the idea that Jordi introduced so well that noise can be especially useful as a bet-hedging mechanism. And so because Jordi introduced the topic so well, I can make my introduction very short, and so I will. And I just want to pose it as this question that organisms have to ask themselves all the time, and the question is, should I do something? And one way to illustrate that is with a seed that's, a seed that's sitting in the ground of a plant, and it has to decide when to germinate. And as biologists, we're all used to thinking about, well, there must be some signaling systems. There must be something that tells the seed when there's enough water, when the temperature is just right, when probably the day length is the right time to come up. And then it's going to make a decision. It's going to send down roots. It's going to send up shoots. And it's going to be a happy little plant. But what I want to talk about today is the other alternative that a seed sitting in the ground has, which might seem like a counterintuitive strategy, but that is for it to not pay attention to very many cues at all, and for it just randomly to send up a shoot and send down roots, okay? And it turns out that under certain circumstances, in particular when the environment is fluctuating, that actually can be the best strategy. And this was shown a long time ago, almost 50 years, in some really beautiful theoretical work by Dan Cohen. And so he built a mathematical model of a seed making this very decision. So here's his model. It's of an annual plant, the seeds of which can either germinate or remain dormant for some amount of time. And what he realized and built the mathematical structure to show is that one obvious way to survive and reproduce in a risky environment is to spread the risk so that one failure will not be decisively harmful. So that's the same advice that a good investment advisor will give you. All right, don't put all your eggs in one basket. That's basically the idea here. I have to point this out. One of my favorite things about this paper is that this is the entire introduction. See, when you have a good idea, it doesn't take a lot of explaining. So one of the best studied cases of bet hedging, this idea of spreading out the risk so that different members of a genetically identical population are doing different things, is antibiotic persistence in E. coli. So this was discovered in the kind of experiment that's shown here. So the idea is if you take a large population of E. coli cells and you expose them to a massive dose of an antibiotic like ampicillin, and you measure how many cells are still around after some amount of time, you get this kind of killing curve. And you can see this is on a logarithmic axis, so 100% of the cells goes down to a very, very small proportion of cells within a few hours of being on ampicillin. But then what you see is that the curve slows down, okay, and the die-off is getting slower at this point. And so one way to interpret this, I think the intuitive way to interpret this, is there was either pre-existing or arising in the population some mutant genotype that's better able to survive on ampicillin, okay? And there's an easy way to test that hypothesis, and that is to take a cell that's alive at the end of the experiment and do the experiment over with its descendants. Because if there was indeed a genetic difference that made this curve slow down, then if you redid the experiment, you should see just slow decline. You would not see the rapid die-off at the beginning. But in fact, when you do this experiment, and this experiment has been done many times, you get exactly the same result. So that shows that it's not a genetic difference here that's causing this. But in fact, as we now know, what's going on is that a small fraction of the population before ampicillin was applied is in a state that's resistant, more resistant to ampicillin. And these so-called persister cells are the ones that are staying alive even as you go many hours out onto ampicillin. And what Natalie Balaban and Stan Leiber showed is how this works at the single cell level, so, or what the phenomenon looks like at the single cell level. So they designed these microfluidic devices where you have these narrow channels. So each of these vertical channels is just wide enough for a single E. coli cell. And they labeled the E. coli cells with GFP, so you could see a streak here where there's a set of E. coli cells that all descended from the same individual. And through this microfluidic device, you can pipe in medium that either has or does not have ampicillin. And so this is what happens when you just start growing these cells without ampicillin present, and you can see that these lineages of cells are growing out. And then they pipe in ampicillin, and you get massive mortality. And then you can take the ampicillin away and see who survived. And what they found was that you do indeed get these surviving cells that can then grow out. And if you trace them back, they were either not growing at all or growing very, very slowly before the stress, okay? And so the idea then is that within this isogenic population, you have the cells that will grow fast because conditions are benign, but there's always set aside a small proportion of cells called persisters that are relatively dormant and that are better able to withstand a stress if it comes along. This kind of phenomenon is not limited to bacteria. So this is one of my favorite recent examples of cancer cells. So these are cells from a melanoma tumor. And what the Herlin lab found is that within that tumor, there's subsets of cells that are either growing fast or growing slowly. And you can see that because they have this proliferation marker, Ki67. So you can see these cells are proliferative, fast dividing cells. But then there are some cells that are not marked by that marker, and they're actually marked by a different marker, which is this JARID1B, which is a histone demethylase. And so this mutually exclusive expression is such that you have fast dividing cells and you have slow dividing cells expressing this marker. The slow dividing cells are more resistant to chemotherapy. And what they found is that if you knock out this gene, the tumor will burn itself out because it does not have this population of slow-growing cells to sort of reseed the population. What's important is that this is not a cancer stem cell, okay? Because a stem cell would be only unidirectional. You go from this pluripotent to some differentiated state. But what they were able to show is that the switching goes in both directions. So you can go from the fast-growing state to a slow-growing state, and you can go from the slow-growing state to the fast-growing state. And that's true of the persister cells in E. coli as well, and a few other examples of cancer cells that have come along in recent years. So we wanted to study this kind of phenomenon, this non-genetic heterogeneity as a bet-hedging mechanism, and we chose to do it in the yeast Saccharomyces cerevisiae. So there are some advantages to doing it in yeast. The first is that yeast are ecologically diverse, so they inhabit a variety of natural and industrial habitats. So out in the wild, if you find Saccharomyces cerevisiae, it's likely to be in the soil around oak trees. But obviously, Saccharomyces cerevisiae is a very important industrial organism that you can find in lots of nice places like breweries and wineries and bakeries. It's also an opportunistic pathogen, so patients with compromised immune systems are subject to Saccharomyces cerevisiae infections. And so this ecological diversity suggests that there might be various strategies out there in the world of budding yeast to deal with various different types of environments. The other main advantage of using yeast is that it's a model eukaryotic species. We have a large amount of genetic and genomic tools that we have at our disposal to study them. And then also, unlike bacterium like E. coli, as a model eukaryotic cell, we have the potential to apply what we learn to other eukaryotic systems, potentially cancer cells. So what we did was to set up a time-lapse, high-throughput microscopy system for following yeast cells as they grow. So this is what it looks like. We have a microscope that has an environmental chamber on it, so we can keep the conditions as we want them for a long time-lapse experiment. Everything's automated about the microscope, so the stage, the fluorescence filters, the camera, so that we can visit lots of places on a microscope plate and go back to the same places over and over and get nice time-lapse movies of lots of colonies growing. So we do it in these 96-well glass bottom plates. We coat the glass with a lectin that the cells stick to, so we can grow them in liquid medium, but they drop to the bottom and stick and grow out in a monolayer. So it's very convenient to set up experiments. It's not microfluidic, so we choose an environment and we stick them in it. But whatever we lose from that, we gain from having massively high throughput. So we can image 30 fields per well in a 96-well plate once per hour, and we get about 30 microcolonies per field. So in one overnight experiment, we can watch almost 100,000 colonies growing. And as I said, we can also, if we choose, use a GFP or other fluorescence marker and correlate expression with growth rate. So this is what one of the movies looks like. So on the left, you'll see just the bright field images, and you can probably already see there are some spots here. Those are single cells that are going to found these colonies. And then this is the software we wrote to track those colonies as they're growing, and you'll see the areas of the colonies are marked. Okay, so the colonies grow out, the software tracks them, the software is smart, so if the colonies run into each other, it stops tracking them instead of saying that the colony instantly tripled in size. And what you can see when I zoom in on just 4 of the colonies growing is the first observation that we made that told us that we were onto something. So these are genetically identical cells, they're clonally related, they're growing in exactly the same environment, they're neighbors. And what you can see is there's a huge difference from colony to colony in the growth rate. So this colony grew very, very slowly. This one grew very, very fast, and there's a few in between. And so, again, the nice thing about our method is that we're not just limited to watching a few movies, but we can get tens of thousands of colonies in one experiment. And so we can get the full distribution of growth rates of these colonies. And the distribution typically looks like this. So we have a big peak around some very high growth rate, and this is comparable to the growth rate we think of for budding yeast, around 90 minutes per division. But they have this really big, thick left-hand tail of slow-growing cells. So in this example, about 10% of the cells are growing at less than half the median growth rate. And this would be a very dumb strategy to have if all you would ever see was benign conditions, right? Because if you're given every nutrient, which we give them, every nutrient they could possibly want, the best strategy is to grow as fast as you possibly can, because otherwise you would get outcompeted by a genotype that was doing that. So what this suggested to us is that there must be some reason that there's this slow subpopulation, and the best reason that we could come up with is that this is bet hedging, that the slow growing cells are at an advantage in some other environment. And so we wanted to test that, and we also wanted to start using yeast genetics to start understanding the mechanism of what might be going on here that differentiates these cells from these cells. So the first thing we did was to try and find some candidate genes, some markers, like what's the yeast version of that Jared1B that might be marking these slow-growing cells. And so what I reasoned was that, well, what would be the properties of such a gene that might be a good one to look at? It would have to be a gene that had some relationship with growth rate, and it would have to be a gene that was variably expressed from cell to cell. And so we had 2 data sets available from other labs that we could use to try and intersect to find our good candidates. And so one of those data sets was from the Botstein lab, and what they had done was to grow yeast cells in a chemostat under different conditions. So these are different nutrient limitations, so increasing dilution rates of glucose-limited media or nitrogen-limited media, phosphorus, and so on. And in a chemostat, of course, you tune the growth rate by the dilution rate, okay? And so what they were looking for was a correlation in gene expression with whatever the growth rate happened to be of those cells that they had set. And they found a large number of genes that correlate with growth, where their expression correlates with growth rate. and those are shown here. So there's some genes that go up in expression as growth rate increases, and there are some genes that go down in expression as growth rate increases. Now, it doesn't necessarily have to be true that a gene that correlates in a population of billions of cells with the growth rate of that population is also going to be important at the single cell level, but that was all we had to go on, so we used it. And then the other data set we were able to use was from Jonathan Weissman's lab, where they had looked at a GFP tagged collection of yeast strains, so thousands of yeast strains with GFP attached to the coding sequence of the protein in the endogenous gene's location. And they had done flow cytometry to look at basically the distribution of fluorescence for each of those proteins. And there are some that came out that were very highly variable from cell to cell. Okay, this is an example of one of those. You can see some bright cells and some dim cells. And so we can intersect those two data sets and try and find ones that show high variability from cell to cell and also show a correlation with growth rate. And so if you plot those two against one another, this is what that looks like. So this is the growth rate correlation. These are genes that have high expression when the cells are growing slowly with a negative correlation. And then this is a measure of expression noise, okay? Higher variability as you go up and lower variability as you go down. And so genes in this quadrant would be very interesting because they both have high expression noise and a high correlation with, negative correlation with growth. And so there are about 80 genes in that quadrant, and when we looked at just overrepresented categories, what we found are two, so ATP synthase and trehalose metabolism. And trehalose metabolism particularly caught our eye. One of the reasons is that there's a trimeric complex that produces trehalose, and TPS1, TPS2, and TSL1, so these are enzymatic components, and this is the so-called regulatory subunit of the complex. We're all in that quadrant, and TSL1 especially was way out here, very negative correlation with growth and a very high variability from cell to cell. And so this is what I just said. It has a high negative correlation with growth rate and high cell-to-cell variability in protein expression. This is our own data just to show you how variable in expression this particular protein is. So this is a TSL1 GFP fusion strain, and this is flow cytometry data, such as a histogram of intensity levels, okay? And this tail goes out pretty far, so this is the same histogram, but you can see it's cut off at 1200, so it's basically the histogram you would get if you just limited it to that. And you can see that some cells have much, much, much higher fluorescence than the majority of cells. The other reason trehalose synthesis was very interesting is that trehalose is thought to protect against stress. So for the chemists in the audience, there you go. It's a disaccharide thought to preserve protein folding. Its levels are known to rise rapidly as yeast cells approach saturation, as they start starving. And in a variety of organisms, it's been associated with resistance to heat, cold, desiccation, oil, ethanol, lots of stresses. And so this is a really nice example of that. This is the so-called resurrection plant, Selaginella lepidophila. And so this is what happens to this plant if you don't water it for a month. This is what happens to every plant if you don't water it for a month. But what's special about this plant is then after that, if you water it again, it springs back to life. Okay, and the reason is that its cells have a very, very high concentration of trehalose. And so now what we want to do is we want to start putting these things together. So we have this difference in growth rates in the colonies. We want to see if that correlates with stress resistance, and we want to see if it correlates with this candidate gene expression, TSL1. So the first thing I'm going to show you is the stress response. And so the way we do this experiment is the same way I introduced to you. So we do the time-lapse imaging, but halfway through we heat shock the plate, and then we continue the experiment so you can see what happens afterwards. So what you're going to see here is a movie. It's going to play. The colonies are going to grow out. It's going to flash heat shock, and then you're going to see what happens. Okay, so there was clearly one winner and 5 losers, right? So this colony survived heat shock, this one didn't, this one didn't, this one didn't, this one didn't, this one didn't. And if I play it again, now you know this one's the winner, okay? What you'll see is that it was very slow growing before the heat shock. Okay, there are only a few cells there, whereas you can see how many cells the other colonies had by the time the heat shock arrived, okay? So again, that's a pattern that holds up statistically when we look at thousands of colonies, right? And we can see it clearly in a movie like this. So the next thing to see is the exact same movie, but over here I'm showing you the channel that's the GFP channel, because these are TSL1 GFP expressing cells, okay? And so this colony, which you now know is the survivor, is going to be somewhere over there. Okay, and what you can see is that before the heat shock, that was the only one expressing TSL1. I'll show you that again. Okay, so we know to look somewhere right there. Okay, TSL1 expression, heat shock, that's the only colony that survives. And then the other thing you notice is that it switches back, right? So the descendants of those cells that are generating this very large colony by the end of the experiment are not expressing TSL1. And we see switching in both directions. So we see TSL1 positive cells give rise to cells that have basically no TSL1 GFP fluorescence, and we see cells that have no GFP fluorescence turning into cells that are expressing GFP, and that can happen with or without stress. So it's, the switching is not dependent on there being stress. And so now that we know that TSL1 GFP seems to correlate with both the slow growth and the stress resistance, we can do a different type of experiment to confirm that. So here's that histogram of TSL1 GFP fluorescence intensities. And so what we can do is to pull out the very brightest of the cells. So this is the 1/1000th brightest cells or the 1/100th brightest cells. And we can ask what their stress sensitivities are. And when we heat shock those cells, the very brightest cells are much better able to survive heat shock than the bulk of the cells. So this is a 30% survival compared to about 7% survival. And it seems to be graded, so if you're in that next best category of the 1/100th brightest cells, then your survival is also elevated above the bulk of the cells, but not quite so much as these cells. So the next thing we can do is ask the question, well, okay, so TSL1 expression correlates with slow growth and it correlates with stress resistance, but is it required for stress resistance? Is it required for the growth? And so to do that, what we can do is a knockout experiment. So we can take a strain that has TSL1 knocked out, or we can take a control strain that has a knockout of some irrelevant gene, so we're dealing with exactly the same genetic background. We grow those cells in logarithmic phase for a really long time so that they have no memory of where they were before, of any history of stress. Then we deliver a very acute heat shock, 2 minutes to 60 degrees. So, and this is very important for bet hedging, right? If you're trying to study bet hedging, what's important is that an organism is prepared for a stress that is coming very acutely right around the corner, okay? It's not that it's mounting a stress response when that stress arrives. It has to be prepared already. And so the best we can do is to do a really, really intense heat shock for a short amount of time so that we're not allowing those cells to actually mount the response. They have to be ready already. And so we do that, and then we do old-fashioned, just plating the cells on agar plates and counting how many colonies survive. And when we do that, what we see is that under these conditions, 5% of the control cells survive, but 2.5% of the TSL1 knockout cells survive. So that suggests that TSL1 is indeed contributing to the stress resistance of these cells. I'm sure it's not the only thing contributing to the stress resistance of these cells, and I'll get back to that. The other thing I should say, which I'll also get back to, is that these TSL1 knockout strain cells do not have a different growth rate distribution from wild type cells. That is, the TSL1 does not appear to have any effect on the growth rate distribution. Okay, so just to summarize so far, what we have is a putative bet hedging system in yeast. So we have wide variation in growth rate among isogenic cells that are grown in the same benign environment. This slow growth correlates with expression of a stress protectant molecule, TSL1 protein, which presumably produces higher levels of trehalose. And it correlates with survival of very acute heat stress. Both states, so the growth state and the TSL1 expression, are transient and switch in both directions. So the next questions we wanted to ask are, well, okay, why? Both in terms of the mechanistic reason, so what is it that's causing these differences in growth rate and stress resistance among cells? So what is the heterogeneity mechanism? And what is it good for in nature? And really the only way to get at that is to start looking at natural strains and seeing if there's differences in their growth rate distributions. So the two things I want to talk about now are our efforts to try and understand the mechanism of heterogeneity and also to look at natural variation in growth strategies among yeast strains. Okay, so on the mechanism side, everything I've told you up till now suggests this very, very simple pathway of this form exists. That there's probably some variable upstream regulator, and then there's a branch point that controls the expression of TSL1 and probably other stress resistance genes, and that controls growth. Okay, so promotes expression of TSL1 and reduces growth. And the reason I say there's a branch point here is because, as I told you, TSL1 expression is required for the stress resistance to the acute stress, but it's not required for the growth rate distribution. So if we knock out TSL1, we get the same growth rate distribution. So there must be some kind of branch point. So we thought what we'd have to do to figure out what the pathway was here is to take advantage of the fact that we could sort out those really intense GFP, TSL1 GFP cells, and then we could do transcriptomics or proteomics and see what else is co-expressed with those, right? And maybe that would tell us what the pathway is. But we were fortunate because someone did that experiment for us. And so Jacob Stewart Ornstein, working with Jonathan Weissman and Hannah Al-Samad, had done an experiment that, it was published in Molecular Cell, so it worked. But if they had asked me beforehand, is this a good project to do? I would have said, no way, okay? I know this is on film, but I'm going to tell you this anyway, right? So here's the deal, if you haven't seen this experiment. So what they reasoned was that if you're interested in identifying members of the same pathway, then one way to do it might be to look for correlated noise. Okay, so if you have something that's fluctuating and something that's fluctuating with it, then either they're both listening to the same upstream factor or one of them is listening to the other one. Okay, and that causes a correlation. And in principle, that's a perfectly good thing to do. But I would have thought that the noise of the experiment would have swamped out the correlated noise that you're trying to find, and it would just be a very weak method for trying to find pathway members. The other thing they did is, if they had asked me, well, what's the right way to set up this experiment? Okay, you can go ahead and do it if you want. But at least make them two different colors. So have a red fluorescent protein and a green fluorescent protein. But instead, it was a green fluorescent protein and a green fluorescent protein that they're trying to find correlations with. And that seems to be kind of setting yourself up for trouble, too. Nonetheless, it worked. Okay, and what was nice for us is that one of the proteins that they were looking at in terms of seeing if it correlated with other things was TSL1. Okay, so by doing their experiment, they can tell me a list of proteins basically that are co-fluctuating with TSL1, and that's what's shown here. So their focal protein was actually PGM2, but then it correlates highly. This is just a score of how correlated it is with PGM2, and so TSL1 scores highly on that axis, and everything else here is also correlating highly with PGM2 and therefore also correlating highly with TSL1. And so here's a list of a bunch of genes, and you can see this axis is response to heat shock. So they turn out to be a bunch of stress response factors that are correlating expression with TSL1, so basically what we would have wanted to find anyway. The other nice thing is that all of these orange dots are corresponding to genes whose upstream regions have at least one binding site for a particular transcription factor, MSN2, which is the canonical stress response transcription factor. And so not only do we have a bunch of things that co-fluctuate with TSL1, but we have our candidate pathway, okay, which is the MSN2-4 stress response pathway. So just a very quick introduction to this pathway. Under benign conditions, cyclic AMP levels are high. E. coli people, don't get confused, okay? Cyclic AMP levels are high under benign conditions, leading to activation of protein kinase A. Protein kinase A phosphorylates MSN2 and keeps it out of the nucleus. Under stressful conditions, cyclic AMP levels are low. pKa is not activated. MSN2 is not phosphorylated and is allowed to enter the nucleus and turn on its target genes, which include many of these stress response factors. And so now we can change this hypothetical pathway to something slightly more specific, but still hypothetical, which is that what's variable might be pKa signaling. And occasionally, even under benign conditions, maybe what's happening is MSN2 and 4 entering the nucleus and turning on stress resistance genes and maybe also turning down growth. So we can test this using genetics. And so the first thing we can look at is whether this pathway is required for these high-expressing TSL1-GFP cells to exist under benign conditions. So what's shown here in white is a strain that has no TSL1-GFP, so this is just basically blank cells. And what we're measuring is the microcolony fluorescence intensity using our microscope assay. So we're going to get some reading, but it's a very low reading. That's just the control. Okay? In the TSL1-GFP cells, what we see is this distribution shifted to the right because you have colonies that are expressing higher levels of the fluorescent protein. And so now we can ask what this distribution looks like in an MSN2 mutant or an MSN4 mutant. MSN2 and 4 are basically partially redundant. So in the TSL1-GFP MSN2 mutant, what you can see is that the fluorescence intensity pattern basically matches as if there's no TSL1-GFP present. So that shows that this variable expression of TSL1-GFP is indeed downstream of MSN2. What's interesting is it's not apparently downstream of MSN4. So when you knock out MSN4, you get basically the same distribution as wild-type cells. Okay? So that suggests that this part is right. At least MSN2 is upstream of TSL1. And as I said, probably other stress resistance genes, although we haven't tested any others yet. So the next thing we want to know is how does it affect growth? And so this is just the growth rate distribution that we see in wild-type cells. And when we knock out either MSN2 or MSN4, what you can see is you get a taller peak and a reduction in this left-hand tail, okay? Suggesting, again, that this is the right pathway, that MSN2 and 4 are upstream of this slow-growing state, that they're required in order to enter that bet-hedging subpopulation. Okay, so now we have that each one of these factors is required to slow growth, and MSN2 is required to activate TSL1 in benign conditions. So the next thing we want to do is put all of these things together, and this is sort of where we are now. We want to look at MSN2 localization in the nucleus at the same time as we're measuring growth rates, at the same time as we're measuring TSL1-GFP expression, at the same time that we can look at the resistance to heat stress. Okay? And so we were lucky because Megan McLean at Princeton had made an MSN2 mCherry reporter, so we can look at nuclear localization. This is what a movie with that reporter looks like, and basically what you're supposed to see is not a whole lot, right? You know, if you look at it at this level, it's not really clear that anything's going on with MSN2. But if we zoom in on one colony, then it starts to get interesting. And what I was hoping was that what we would see in, say, a slow-growing colony is MSN2 would just be sitting in the nucleus. Okay, it would be really obvious that that cell is somehow activating that pathway. That's not the case, apparently. And Jordy won't be surprised by this, but what's actually happening is MSN2 is shuttling in and out of the nucleus. And probably what we need to be able to record is the dynamics of that fluctuation, and to understand what statistics of that, whether it's amplitude, period, whatever, is correlating with the slow growth state. And so we're not there yet in terms of extracting all the data, but I just want to show you that we can see such things. So this is just the bright field image of this one colony growing. the MSN2 cherry signal, and then the merged image. And what you'll see is these little pops of, this is a 5-minute time scale. You'll see these little pops of MSN2 in the nucleus. Okay, it's going to repeat again. And if you keep your eye on this cell that forms here, this new bud, you could just get this flash in the nucleus. So this tells us a few things. One is we really have to be careful here. This is not going to be an easy problem to decipher what's important in terms of making a colony grow slowly. We probably also have to do a much finer time scale. If this shows as a single frame flash in this movie, then 5 minutes isn't short enough to actually capture the relevant dynamics, and we're going to have to go to a much finer time scale. But we're hoping that we'll be able to do that. Okay, so to finish up, I just want to let you know where we are in terms of looking at natural variation in growth strategies, because I think ultimately this is what we want to know. Do changes in the distribution of growth rate and stress resistance matter in nature? And so Naomi Ziv, who's a really fantastic PhD student who convinced my colleague David Gresham and I both to be her PhD advisors and then created her own project to make that happen, generated data on a bunch of wild isolates of Saccharomyces cerevisiae, and she used our growth rate assay to look at their average growth rate and their variance in growth rate. And so what you can see from this plot is that there's a wide variation among wild yeast strains in how fast they grow and in how much variance there is in their growth rate. So you can see some that have very high average growth rate and very low variance, some that have very low average growth rate and low variance, and you see other things in between. One of the most interesting pairs to us was these two strains, because they have the exact same average growth rate, but they clearly differ in their variances. And you can see that, I hope, a little bit better here in these two histograms. So you can see that this strain from the Netherlands has just a wider distribution than the strain from Finland. Okay, and these are both they're very closely related genetically, both from Northern European soil. Okay, and so we're hoping that we can map the genes underlying this difference. What is it in these strains? Which alleles are causing this difference in variance? Okay, and we might have to become ecologists to really understand what's going on, but I think at least we can do the genetics. The one thing I should say is, and people encounter this with various wild strains of yeast all the time, is that these two strains are hard to do genetics with. They don't like mating with each other. They give very low spore viability. Some people are very interested in that as a trait. You know, people are interested in speciation. We're not quite interested in that. We see it as a nuisance because we get very few progeny out of our crosses. And if you're trying to do quantitative genetics, you need a lot of progeny. So we're not there yet in terms of using these strains as parents to map loci underlying a variance difference in nature. But in the meantime, we've used a set of strains that Barak Cohen created, which is a cross between a yeast strain from the soil around an oak tree and a vineyard strain. They made 374 F2 progeny strains, and they genotyped all of them. So all we have to do is measure our phenotype in those strains, and then we can do all the mapping for free. And so we have measured the growth rate distributions of each of these strains, or Naomi has, and what she sees is she can map quantitative trait loci for both the mean growth rate. Okay, so this is a LOD score plot with all the yeast chromosomes showing, you know, some pretty major peaks that determine a difference in growth rate average between strains, and then she also can map quantitative trait loci for just the variance. And what you see is this peak here, for example, is a QTL for variance, and there's no corresponding QTL for mean. Okay, so that's a allelic difference in these natural strains that controls variance alone. So we're pretty excited about both of these directions. We're hoping that we can really nail down how this pathway is controlling this heterogeneity mechanism. And we're hoping by using this complementary strategy of looking at natural variation, which we expect will also turn up genes involved in controlling variation and growth, that we'll have a much deeper understanding of a real bet hedging strategy and what it might be doing in nature. So with that, I want to thank a few people. So Sasha Levy was a postdoc in the lab, really, really wonderful, wonderful scientist who did most of the work I presented today. He's starting his lab at Stony Brook University, basically today. Here's Naomi, who did the work on the natural variation with David Gresham. And Shuang Li has been doing the work on understanding the genetics of the PKA pathway, MSN2. These are the rest of the people in my lab, half of whom are undergraduates who are really fun to work with, former members of the lab, wonderful colleagues at NYU, and people who pay the bills. And thanks for your attention.